Thank you all for joining us. I'm very pleased today to have the opportunity to introduce to you Dr. Fred Andrus. Dr. Andrus comes from the University of Alabama, where he is a professor and chair of the Department of Geological Sciences. He did his graduate work at the University of Georgia and has done a lot of different types of work, primarily looking at paleoproxies, which is what he's going to talk to us about today. So thank you.
Thank you, Ruth. Let me start off with an apology on a couple of counts. One, I'm battling allergies, so I'm going to go into periodic coughing fits, and my voice is going to give out, so forgive me that. Then also forgive me because I probably don't really fully fit in at Dauphin Island. I do marine science of a sort, and you might think what's next out of my mouth is, but I'm really a geologist. I mean, I'm actually not even really a geologist. My background is primarily actually in archaeology, and my principal research interest is in human-environment interactions over deep time. Thinking like early civilizations, how did people learn to make a living, particularly in coastal areas? And what that led me to do was study midden remains, oftentimes shellfish, but sometimes vertebrate remains found in archaeological sites around the world. And it gives me insight into a couple of things. Like a lot of what I'll be talking about today is the chemistry of skeletons and how that can teach us something about ancient climate environments and ecology. But then also I'm interested in people and the way we have adapted to living, particularly in coastal environments, often over seasonal timescales. When I was formulating the talk, Ruth was kind enough to invite me. I've been here before, and last time I spoke here, I did a lot about archaeology. And so I'm going to actively try and avoid it this time. There's a little bit of archaeology in the talk, but instead I'm going to be mostly focusing on the if you will, the P-cubed area of geology, paleoecology, paleontology, and paleoclimatology, particularly dealing with sclerochronology, which is this sort of $6 word here that some of you may be familiar with, others may not. I should apologize for a third thing. Also, I've never given these data publicly before. So everything you're seeing I've never presented, I've never spoken. So this is not going to be a particularly smooth, slick talk. And because this probably isn't necessarily in your subject area, please interrupt me as I go, because I'm probably going to skip over things that are important to you. Let me know, and I'll be happy to jump in there. I want to give credit where it's due, though, and much of what I'm going to be saying today is owed to these folks here. Christine Bassett is a present PhD student of mine. She also did a master's work with me. She and I just actually returned from the Aleutian Islands like 3 weeks ago, and we'll talk about our work up there with butter clams, Saxodomus gigantea. I also want to give a lot of credit to a former PhD student of mine, Hillary Slutton. She's actually now at Shell Oil, but she did a great deal of work talking about ocean acidification and the effects on coralline algae, and also using coralline algae as a paleoproxy, and that's what I'm going to focus on today. And then finally, a professor at William & Mary and her student Josh Zimt, who's now at Berkeley, dealing with ancient oysters. She's had some really fascinating studies published recently talking about the evolution of oysters along the east coast of the U.S. So this is going to be 100,000 to a couple million-year-old oysters, same species that we eat today. So it should be, hopefully, to you kind of interesting. But I want to give credit for them and a lot of my other colleagues along the way. So first of all, let's talk about what sclerochronology means. And this is a word very similar to dendrochronology. So dendrochronology, tree chronology, tree time. This is skeleton time. And the word was coined, I think, focusing primarily on corals and the way you can interpret the growth rates of corals or the geochemistry of the coral skeleton to interpret ancient climate. It seems kind of esoteric, and usually when I explain what I do to a non-scientist, it seems almost pointless. It seems so disconnected from any sort of meaningful impact on the world. But I would argue it is very impactful, that if you think about what we know about ancient climates, this is primarily it. It comes from trees, marine skeletons, and I'll talk about why that's really important, and ice. And those three prongs tell us almost everything we know about global warming and give us the context we understand about anthropogenic global warming. And to best build these proxies, these indirect measurements of past climate, we first must at least pretend to be marine biologists, at least if you're working on the marine skeletons, just like dendrochronologists must at least pretend to be plant scientists. The reason geologists focus so much on skeletons is because this is one of a handful of minerals that grow on the surface of the earth. So geology is primarily a forensic science like archaeology. We're trying to figure out things we can't see or measure directly. Maybe it's inside a volcano or at the core of the earth or something that happened 100 million years ago. We need something durable that is a product of its environment to be able to figure out what happened when we can't see. Most things that the planet is made out of were produced deep in the earth, and they're great for understanding how the deep earth operates, but they're terrible at understanding how the surface of the earth operates. So if you could really come up with a short list of the minerals that grow on the surface of the earth, by far and away the most important is going to be calcium carbonate. And it comes in primarily two forms, aragonite and calcite. And it's insanely abundant over the past 500 million years on Earth. You have older examples, but at least for the past 500 million or so years, this is super, super common. And it's still the main thing that marine critters make their skeletons out of today. A few exceptions like diatoms, but this is pretty much it. And when you look at these skeletons, unlike our skeletons, they're more or less constantly being reworked, unless you have osteoporosis. These skeletons grow on themselves. They have an internal stratigraphy, at least from a geological perspective, stratigraphy. And just like in a tree ring, you can count back years if the tree grew in a place that has seasons. You can do this with a wide cross-section of marine organisms. This is a limpet, snook otoliths. Otoliths, in fact, probably somewhere where most of you have done sclerochronology, even though you might not use that term. This is sclerosponge, coral, which a lot of the coral growth bands are actually only evident in X-ray density features, but they're there. And this is a beast that's really important geologically, but not so important biologically now, brachiopods. And if you go back to the Cambrian, they were just insanely common, although they're just about gone now. Pretty much everything that makes a skeleton makes something that can be interpreted for time. But if you're going to try and analyze these skeletons to figure out something about the past, ecology, biology, climate, you need to figure out the calendar that it records. And there's a big spectrum of time scales at play. This is a colleague of mine, Al Wanamaker. He's at Iowa State. He used to be at University of Maine and Wales. And he works on one of the longest lived organisms on Earth, Arctic Icelandica. And Arctica live 550 years. It's a clam common in the North Atlantic. And they live in big groups. And much like with tree rings, you can cross-date the growth structures in these clams and create chronologies that are several thousand years. And they live in a climatologically interesting place, the North Atlantic. It's really key for understanding how the last few ice age cycles have happened. So you can get really, really long-lived records from the ocean. Corals, depicted here, they also can live for several hundred years. Those are go-to proxies, if you will, for understanding past climate, because you can get measurements of maybe growth rate or geochemistry for long sweeps of time. So you can detect not only relatively short events like seasons or El Nino, but long-scale events involving orbital patterns of the Earth. Gives you context for understanding global warming. Now, within these lifetimes, you're going to see many different subdivisions of time. Famously, corals, they make a little growth line every morning, at least if they have photosynthetic symbionts in their skeletons. Also, this is a clam I'm going to be speaking of later, Saxodomus gigantea, the butter clam, really common on the west coast of the U.S. They make a growth increment every tide. We call them lunar daily bands. Many organisms do it. Rangia cuneata do them right here in Mobile Bay. I have one paper, we're looking at that. So you can study the time scales of multiple thousand years down to a few hours, depending on the organism. And if you're really, really lucky, the holy grail is to get something that lives 1,000 years, but you can sample at maybe daily or weekly time intervals. Very rare, but it does exist. So, this is not new in geology. In fact, this is kind of bedrock geology. If I had to name one of the most, probably is the most famous paleoclimate paper, it would be this one in Science, Hayes, Embry, and Shackleton, when they used the skeletal chemistry of foraminifera to confirm that Milankovic was largely right. And it governs our understanding of how ice ages come and go over tens to hundreds of thousand year cyclicities. And the reason this is possible is this mineral grows with a particular isotope chemistry that enables us to either calculate temperature or the isotope composition of seawater. And in the case of Hayes, Embry, and Shackleton, they were dealing with deep ocean benthic foraminifera extracted from cores. And they knew that the temperature was relatively constant over these tens of thousands of years that the cores were deposited. And therefore, they could measure isotope chemistry up and down the core and track global ice volume. So it seems very disconnected. A microorganism on the bottom of the ocean tracking our geometric relationship to the sun and how it changes over hundreds of thousands of years. But believe it or not, you can make that measurement. Nowadays, I would say coral are the canonical marine proxy that has seasonal timescales. Foraminifera are great, but they don't live very long. They die young. They settle into the seafloor. They get bioturbated. So you end up with a very coarse understanding of how climate changes through time. Whereas a coral grows much more rhythmically, and we can understand that rhythm very easily. And here you can get seasonal or even sub-seasonal resolution of environment. The basis of this is the chemistry of the skeleton. I'm going to go into that. It's way more than I can talk about today, so it's just going to be, I'm just going to sort of dip my beak in the water a little bit, give you some introduction to the chemistry of these skeletons. It does have some applications in modern species, and so if you have any questions or if you catch a mistake I'm making, and gosh, I know I'm making them, interject, and we can talk about some of these other ways this can be applied. Why is this so important, though, for us? This is a great plot from a PAGES conference. This is a global change organization. I've been to one of their conferences. These are really great workshops where you focus on wrestling with big problems in the paleoclimate world. And one of the biggest problems we have is an incomplete temporal and geographic availability of paleoclimate data. And if you look at this plot, this is maybe the most telling. This, broken down by the different proxy sources, green being dendroclimatology, this shows how far back in time. And you see we lose most of our tree records pretty quickly. In fact, most terrestrial records, with the exception of maybe lake basins, disappear fairly quickly. What we're left with in deeper time are almost exclusively marine records of climate change. And that's great. The oceans have a huge amount to do with understanding the climate system. But if you look at them, they come really in two general categories. The very coarse, poorly temporally resolved records, like from sediment cores. Those are wonderful for understanding how climate changes over millions of years, but they're terrible for understanding how climate changes over hundreds or thousands of years. So then we look for seasonal or subannual proxies for that. The best subannual proxy, like I mentioned, is coral. So if you look at the distribution of proxies, they're wildly uneven. So the coral records here depicted in orange, of course, we're confined to the tropics. We don't have an equivalent proxy elsewhere in the world like coral, something that lives hundreds of years, has great chemistry, and can tell us all sorts of great things about climate. So the big emphasis in my community over the past 20 years has tried to come up with alternatives. What other marine organisms make skeletons that persist either in the rock record or the archaeological record long enough to be valuable? And can we understand them in the modern sense well enough to interpret their chemistry? And that's what I'm going to be talking about today, is this concept of validation. And what we're trying to do is understand the processes that govern the chemistry of these skeletons. I'm going to walk through some of it right now. By the way, I have a clam depicted. You could pretty much put any biocarbonate you feel like here. I just thought this was a pretty picture. So if you go back, the touchstone moment in paleoclimate studies was probably when Harold Urey wrote this paper called Thermodynamic Properties of Isotopic Substances. Now, you might know Harold Urey from the Miller-Urey experiment, where the origins of life and all that sort of cool stuff. But he was a polymath, a brilliant man, and he came up with the idea that stable isotopes distributions can be used for a variety of purposes, one of which would be paleothermometry. So in his paper, he argued how the oxygen isotope distributions in calcium carbonate are a function of two variables: temperature and whatever reservoir of oxygen isotopes the skeleton was growing from. In the case of marine organisms, ambient ocean water. It's a nasty system because you got these two variables, and the deeper time you go, you can't control for both of them, and I'll talk about why that's such a big problem. But nevertheless, this was the best tool we have for understanding ancient climate. Absolutely game-changing. And the very first organisms that he actually started studying after he came up with this was clams, and then it moved on to other organisms. Carbon isotopes are also measured, and he did describe the thermodynamic properties of carbon isotopes, but they're heavily overprinted by biological activity. And so keen interest to biologists advanced ecologists, less so to paleoclimatologists. I will talk about it a bit. But since that initial sort of oxygen-carbon start, now we've got this wide range of potential proxies, chemical proxies we can measure in these shells. I don't study all of them. Let me jump ahead. The blue ones are the ones I'm actually deeply familiar with and I use a lot. Pardon me if this gets boring, you know, step on your foot and wake up. I'm going to try and walk you through a few of these proxies. If we look here, the key thing to understand, particularly about oxygen isotopes, is we have a firm theoretical understanding of why oxygen isotopes vary in carbonates. We're not guessing, we know. And we can prove this empirically. Some of these other proxies are sloppier than that. That we have empirical relationships between some sort of environmental proxy we care about, but we don't necessarily have a thorough chemical understanding of why they behave the way they do. And that's what these validation studies do. The validation studies give us guidance into in what way these different chemical proxies can tell us something about the world and how trustworthy that is. So if we jump over here to elemental chemistry, I don't do a great deal of elemental chemistry. I'll talk a little bit about magnesium today. Elemental chemistry is one of the big frustrations in carbonate work, because if you talk to a straight geochemist who does not work with critters, there's a strong thermodynamic basis for expecting these things to substitute for calcium at a known rate according to temperature. But critters didn't get the memo, and they don't follow the thermodynamic predicted pathway. That's not to say these aren't meaningful proxies of things. They are. But you can't just go to an old textbook in thermodynamics and make sense of what you're going to see. You've got to go measure it in the modern and come up with correlations, and better yet, an explanation of why things vary the way they do. And these can be used for all sorts of things. You know, most famously like otolith natal homing, because these elements will plug into the calcium in these molecules according to ambient abundance. This can be used for anthropogenic loading of different metals and estuaries. That's really great. What guys like me want, we want this to be a temperature or salinity proxy or something that's going to tell us something about climate. And in some cases, it does so spectacularly. In other cases, it fails miserably, but tricks us for about 10 years. More than once, someone says, oh, I've got a temperature relationship between element X and calcium. This is it. I'm going to get my Nobel Prize. And then 3 years later, somebody says, no, you know, it only works under these narrow circumstances, and you're actually misinterpreting the real causes. We can't trust it, and it all dies. So over and over and over, element chemistry is really tough if you want to try and figure out paleo temperatures. So the big breakthrough lately has been this, clumped isotope thermometry. This is mostly the brainchild of John Eiler at Caltech. And he has, based again on Urey's work, developed the ability to measure the affinity for the heavy isotopes of oxygen, the heavy isotopes of carbon to bond to one another. And as they bond, that's a function of temperature. And this is critical. It's wholly independent of the environment in which the mineral grew. So this is a pure temperature proxy. This is the holy grail we've been looking for in paleoclimatology. The bad news is you need really big samples and your imprecision is bad. So you got like that 2 degree plus or minus on those measurements. That's not very good for paleoclimate work. I mean, it's fine if you're worried about something that happened 500,000 years ago, but if you're trying to understand something about anthropogenic global warming, 2 degrees is too sloppy. We're probably going to get somewhere, but this is probably the biggest breakthrough in geochemistry in my career. It'd be interesting to see where it goes. We have isotopes of calcium. No one's found a good use for them. Radiocarbon is what I actually spoke of last time I was here. And radiocarbon is a wonderful proxy of current movement, particularly vertical movement, because radiocarbon is produced in the atmosphere and works its way downward through the ocean basin. So if you're in an upwelling zone or a downwelling zone, you're getting very different radiocarbon signatures that tell you about local currents. Here's what I'm most excited about. And I'm not going to talk about it, though, because you've got someone who knows a ton more than I do about this, which is the chemistry of the organic matrix. Geologists actively avoided talking about the organic matrix for about 50 years. We would dismiss it as a small component of the skeleton, whatever. That's just, you know, like derisively people call oysters snot on a rock. That's the snot. Let's just wash that off and let's focus on the rock. Now, though, we're realizing that you can get a lot of good data out of the organic matrix, and shockingly, it lasts a really long time. We've got organic matrix from the Cretaceous. from mollusks. And I've got colleagues who've measured this from brachiopods back about 200, 300 million years. So organic matrix will last a really long time and it gets trapped inside the crystals. And I think the biggest breakthrough here has actually been stable isotopes of nitrogen. I think it was Stephen Macko's students was the first to do it, but that paper kind of sat on the shelf actually until Ruth picked it up and ran with it, and I think did some really fascinating work with how you can track anthropogenic impacts to ecosystems here. So I am trying to pick up the baton and run with that and apply it to archaeology. But I'm not going to talk about that today. What's the bottom line? That this is, if you were to pick amongst almost any mineral in the mineral kingdom, arguably the best geochemically for trying to figure out this vast array of interesting variables, interesting at least to paleoclimatologists. Again, I'm only going to be able to touch on the processes here. I'm assuming you know what isotopes are. I'm assuming you know a little bit about fractionation. If I'm not right, jump in, I'll try and explain it. But if you've got that general background, the main thing I'm trying to convey to here is this is a Rosetta Stone, but we don't know how to read it very well yet. Now, why is this important? Well, if we can learn to read a wide array of species from a wide array of environments, from a wide array of time periods, we can finally answer some of the biggest questions in paleoclimatology. So when we get away from being dependent on tropical organisms like coral, we can look at the more climatologically interesting places, because actually the tropics are pretty boring climatologically. The action's going to be up at the high latitudes. And if we can get out there, get really throughout what we call the entire Phanerozoic and get a detailed understanding of what drives climate through time. And also because we have a single molecule that is found in all these different species in different environments, it's a multi-proxy. So every proxy's got its problems. This way the problems can correct themselves by coming at it from multiple different angles. One thing though I typically have to convey to geologists and modern scientists to some degree is how common fossils are. I think a lot of people have this perception that fossils are this sort of rare thing, and therefore everything I'm telling you is so esoteric we really could never meaningfully apply to the ancient. Alabama in particular, I petition you to dig a hole south of Birmingham and not find a fossil in it. It's absolutely chock full of them. Most of them are shallow marine, a few deep marine. What about archaeological sites? Well, there's a giant shell midden within walking distance of right here. I would almost bet anything that if I dug a hole underneath this building, I'm going to find a shell midden. They're everywhere. And humans have been eating shellfish since the Pleistocene. So we can get human-sampled marine organisms for tens to maybe even hundreds of thousands of years if you're talking about Africa, and really tell some fascinating stories. So first we're going to learn to validate these proxies. That's the key thing in the modern organisms, because we've got a huge suite of problems with each and every one of these organisms, like challenges we need to overcome before we can interpret them meaningfully. But if we do, the payoff can be really huge. It can be really huge. So what I'm going to do for the rest of the talk is give you three really brief, shallow examples, little 10-minute, three 10-minute mini talks, if you will, about different ways I'm applying this other than archaeology. If you look at my CV, probably 75% of my papers have to do with archaeology, so I'm actively avoiding them today. I apologize about that, but I figured you guys probably didn't care about it. There's a few things that cross-cut all of these talks is the basic methods I'm dealing with. So whatever critter I'm dealing with, this is a Mercenaria clam here. One way or another, I cut it such to expose its ontogenetic growth. And you're probably more familiar with this than me, but basically you can learn to read, like in this Mercenaria here, this would have been shell precipitated near the birth of the organism, and this would have been near death. In some cases, you have obvious growth, like these are most likely annuli in this Mercenaria. And then on one device or another, either a micromill or a laser, we will subsample these across ontogeny. We do this in most of these talks, stable isotopes. Let me go back. And this is my stable isotope lab at UA, and we've done a little bit of elemental chemistry at University of Toronto and some other places. And what we end up with are data profiles of one proxy or another. And what I'm looking for, because I'm really interested in seasonal climate change, are sinusoids. So like this sinusoid actually came from a different Mercenaria, but this is really commonly what you would see if you measure oxygen isotopes through a clam. Maybe before I get here, I need to explain one more thing about oxygen isotopes. Show of hands, how many of you know what they are? Okay, a few. All right, cool, good deal. All right, so the basic idea when you're interpreting these is water is the reservoir from which these organisms build their skeleton. But water isotopes vary spatially and temporally. So when we used to have vast ice sheets on the Earth, a lot of seawater was locked up there. And it was locked up there in the form of snow. And snow, of course, like all precipitation, evaporates from the sea surface. And light isotopes evaporate more easily. So those light isotopes concentrate in the clouds, and they concentrate in the precipitation. And in a place like Greenland or Antarctica, where snow falls but doesn't really readily melt, we're sequestering light isotopes there. So if we melt those ice sheets, we're going to change global ocean chemistry. If we grow a bigger ice sheet, we're going to change global ocean chemistry. And that was the basis of Hayes, Embry, and Shackleton's testing of Milankovich's theories of how ice sheets come and go. If we assume, though, we're dealing with a slice of geologic time in which we understand ocean chemistry, and we're in a place that's way far away from continents and no big rivers, like imagine Bermuda like 2,000-3,000 years ago, then we can solve for temperature. But the relationship is inverse. So when you see a more positive value like this, it looks like higher isotope values, that's actually winter, everything else being held constant. And then this in this clan would be summer. So if you understand and can hold constant oxygen isotopes in the water, there's an inverse relationship between these values and temperature. We're going to come back to that over and over and over again in multiple of these proxies. So let me give you the first bit of this talk. And this is the one I'm most nervous about, because I'm going to bet half of you know more about algae. In fact, I bet half of you have forgotten more about algae than I'm ever going to know. I don't know much about algae, but I had this great student, Hillary Sletton. She needed a dissertation topic. And I've been recently hearing talks by this guy named Jochen Halfar at University of Toronto. And Jochen, along with Walter Addy at the Smithsonian, has been promoting this idea that coralline algae may be the high latitude equivalent to corals. That here we've got a carbonate producing organism that'll live for centuries in climatologically interesting places, and they have really, really simple chemistry. So if you want to get into this, he had a paper in geology. He and several colleagues had a paper in geology where they sort of outlined the approach to this proxy. And the talks blew me away. But to be honest, very few people are picking up the idea and running with it. So we're still learning a lot about the basics of how coralline algae proxies can work, but I'm really excited what I've seen. And if you go to cool temperate places, almost any hard substrate's covered with the stuff. It's everywhere. And it lives for a really long time. And that's enough to make it useful. So we've done several studies in coralline algae. The one I'm going to focus on today is in Greenland. And our question is, can we use the chemistry of these algae to understand deglaciation over the past 100 years, ideally, eventually the past 1,000 years? And the basic rationale is kind of like what I mentioned earlier, that glaciers tend to have lots of light isotopes in them, so they have really negative isotope values. The ocean water has heavier isotope values because that evaporation leaves the heavier isotopes behind. That's the basic rationale behind this proxy. This is a chunk of, or the original chunk from which our slices came from the coast of Greenland. This is the incremental growth. We have in this sample that we've been studying, this is the sample where most of my data are going to come from, is about 100 years. But we weren't able to quite sample the full 100 years because of some bio-erosion that had taken place. How do we know it's 100 years? This was collected live, and at least at this high latitude, the algae don't grow in the winter. So they leave behind a line, and then they produce a new generation of cells in later years. Jochen has found examples that live over 1,000 years in one place, and then we find fossil examples. They concentrate uranium really well, so they're super easy to age date, and we see older examples in the rock record. The species is Clathromorphum compactum. Like I said, this is one of the long-lived species. There's many different species, and Jochen is the one, Jochen and some colleagues were the ones who collected the one we're going to be talking about today. Here's where it came from. This is near the largest quote-unquote city in Greenland. And this is in a fjord that is fed by glacial melt. The glacial ice here has values like -15 per mil, whereas the ocean values is 0 per mil. So shockingly different values. And they mix through this fjord. And so our idea was, if we could somehow control for temperature over time, that we could back calculate the relative contribution of seawater versus meltwater in these fjords and see if it changed. So the way we control temperature is one of these to me, less robust proxies, magnesium, calcium records in here. So we did a laser transect, actually multiple laser transects across here, measuring magnesium calcium ratios. This was actually a Jochen student at Toronto who did this, and her final conclusions were there was very little variation over the life of this organism. In other words, it didn't seem to experience much temperature variation, which is not too surprising because we don't see much difference in the marine communities over this time period either. So basically we can get this and assume constant water temperature. This is one of a number of equations that describe the thermodynamic relationship between oxygen isotopes in a carbonate, the oxygen isotopes in water, and temperature. So if we can control for temperature with magnesium, calcium, we measure what's in the carbonate, and we can solve for the delta 18O of the water, which is a function of that in-member system, where you got the really negative ice and you got the really positive ocean water, and they're going to track with salinity, basically. We measured almost 700 and some odd samples through the life of this organism. This is ultra fine resolution sampling. It took forever. It was just jaw-dropping work on Hillary's part, and we were really shocked to see this negative trend through ontogeny, way more negative than we had anticipated. Again, this is showing the distribution of the oxygen isotopes in global precipitation. So the ice records here, we're talking about, the negative teens per mil in that part of Greenland. And the seawater, of course, is approximating 0. Now, if you notice, I had a asterisk back there on the magnesium calcium. The reason is if you read Hillary's dissertation, a couple papers that came out of it, you'll see that the magnesium calcium proxy doesn't behave itself thermodynamically. And we studied this in Rhodolis in Panama, and where we set up a mesocosm experiment with this cousin. It's not at all the same algae, but it's a cousin algae. And we measured magnesium here in the black line across ontogeny of these algae. And we found this beautiful oscillation. I think anyone in the literature said, oh, that's definitely a seasonal oscillation in magnesium calcium. Look at these beautiful growth increments. So this is a multiple year record of seasonal growth. Since this is Panama, west coast of Panama, we're probably talking about upwelling variation. So these are upwelling years, and magnesium calcium is varying with temperature. Bull. This grew in 7 months in a tank, and it produced these bands that sure as heck look like annuli, that anybody in nature would have seen annuli, but they're not annuli. And each different rhodolith grew a different amount. They each seem to be living by their own rhythm, and they each had these beautiful sinusoidal magnesium calcium variations, which most would have interpreted to be temperature. There's a paper about to come out I'm privy to that will explain this, and it's got to do with the biomineralization of different types of cell walls in plants. And what happens is the internal structure of the algae, as it goes through time, it changes the shell wall structure, which necessarily changes the magnesium composition of the carbonate. It's got zilch to do with temperature, except perhaps temperature in cases may govern cell wall structure, which then governs magnesium concentrations. In other words, it could be an indirect proxy of temperature. But either way, I'm taking with a little bit of a grain of salt that the magnesium calcium record's trustworthy. To be honest, I don't even really need it, because for this to be explained solely by temperature would be about a 6-degree change, a 5 to 6 degree change in fjord temperature, which would be catastrophic. That level of warming just is not a tenable explanation. So instead, what we're seeing here is over the past 100 years, increased input of Greenland meltwater. Further bolstering this is we do have, from written records and satellite records, evidence of melt pulses, extremely hot summers that sent a big slug of water into the fjord. And these boxes each mark one of those big melt events. Now, they're not all perfect, but like this melt event quickly followed by a big negative excursion. Melt event, negative excursion, melt event, negative excursion, again here, gets a little uglier in here, and the temporaneity starts to go wrong. But I'm thoroughly convinced what we're looking at is increased meltwater input over the past 100 years. If you look at the underlying data, here we just have a best fit line. If you did a more complex analysis of this, you can get a sense of different rates of meltwater over the past 100 years. So this paper's not out. You're the first people to see these data, really, except for the people on Hillary's committee. We're going to submit it soon. it's not really shocking to think that there's been anthropogenic warming over the past 90 years. That's not going to turn any heads. What we just wanted to prove is that this new proxy is meaningful for this measurement. And now we want to go find some of these that are maybe 500, 600 years old, and then we can add a meaningful chapter to our understanding of Greenland melt. So it's one of the reasons I'm actually very excited about coralline algae, is I think we can ask detailed questions we haven't been able to ask at high latitudes before based on marine proxies. And this is where I'm going to get into a little bit of archaeology, changing gears. So I just came back from here on Alaska. I had a beard, by the way. I don't know if you'd have laughed at me, Ruth. I actually grew my first beard since I did a 2-week research cruise. I grew a beard on that because I didn't want to cut my face on the ship. And now I grew a beard because when you get to Alaska, and some of you have probably been to Dutch Harbor, I think they confiscate your razor. They just expect you to look a certain way. And so we were around Dutch Harbor, but rather than going for all the glamorous species that you hear about up there, we were going for the lowly old butter clam. I don't know how familiar you are with butter clams. Supposedly they're really tasty, but everyone tries to not eat them because they tend to kill you. They concentrate saxitoxin, and in certain parts of the world, this being one of them, you're taking your life in your hands if you eat them. I found one guy on the island that eats them, and of course he had to be a geologist, which is embarrassing. He's also the radio DJ in town. But as a consequence, no one really seeks these clams out. The reason we're seeking them out is because ancient people used to eat them all the time. And we have middens with these things that are meters and meters deep, and they can help us address an interesting question about climate change in the Bering Sea over the past 5,000 years. So a controversial series of papers came out not so long ago arguing that you had sea ice as far south as on Alaska about 4,000 to 6,000 years ago in what we call the mid-Holocene climatic optimum. It's kind of counterintuitive. That's usually the period when we consider the last 10,000 years was the warmest. Yet we have evidence of sea ice here. What's the evidence? Believe it or not, it's the distribution of seal pups from species that have to calve or to have their pups on sea ice, particularly ring seals. And a few others. There's also, believe it or not, in some of these shell middens, some polar bear, supposedly. Trust me, there's no bear of any type whatsoever on this part of the Aleutians, much less polar bear. I suspect that's probably a trade item. Ring seals, though, probably not a trade item because there's so many other kinds of seals you could eat. This is just food for them. But me and my colleagues, this is Catherine West at Boston University and Mike Utnier at Western Washington, think they're wrong. That this is probably a misidentification, and there was no sea ice down in this area at this time. So at NSF, we convinced them to fund us, and my part of the research is looking at butter clams. And this is one of those great clams that lays down tidal increments. They've been studied actually fairly extensively. Dave Gillikin was the first person to do it. I know you know Dave, Ruth. And Dave was the first one to study this species. He didn't really do as much with it as the Germans have. And there's a German group led by Bernd Schona and his student Nadine Hallman who've done spectacular work where they can literally go back, and this is a living clam from the west coast of Canada, and go back and pin each growth increment to a phase of the lunar cycle based on tidal magnitude. It's spectacularly elegant work. And my interest was, can this be used as a paleoclimate proxy? Because if we would have had seasonal sea ice back then, one would presume we had a significant change in seasonal sea surface temperatures relative to today. And so what Bernd and his students had shown is that in places where these shells don't grow in the winter, you see this really clear winter growth line. In fact, you don't even need to look at it in a microscope. You can just see it on the exterior surface of the clam. These guys shut down in winter, basically. They don't grow. So then between winters, you can count how many tides there's been. Now, there's going to be noise because these clams, Pycnopodii are always trying to eat them. Sea otters are trying to eat them. There's a lot of reasons why they might stop growing and stress out for a while. But generally, in a population of clams, they've been measured like in Dundas Island and a few other key places in Canada, they seem to have more or less the same number of what we call tidal daily increments, or lunar daily increments, sorry, between these winter growth bands. Now you'll get tricked. You'll get other growth bands that aren't winter. So we have to use oxygen isotopes to confirm, based on temperature, that these are in fact winter growth cessations. If we find one that's non-winter growth cessation, we discard that clam from the record. So what my student Christine did for her master's research was take acetate peels, just hundreds and hundreds of acetate peels from clams all up and down the west coast of Canada, all the way up to Alaska. And we're trying to confirm this idea that as we go to places with shorter growing seasons, we're going to have fewer and fewer increments. And there's a quantitative relationship between the length of the growing season and the number of these lunar daily increments in the clams. And you can kind of see this. So if you get down south, like this is not so far from Vancouver, it's really difficult to even spot the winter growth bands because these clams grow fairly happily throughout the year. And contrast that where these Alaskan clams, where you get these sort of sharp, obvious insults really evident on the surface of the clam. And we can go back and measure these isotopes and confirm whether or not that was winter. So we zoom in on the second year of life typically. And the reason we do that is these things, the growth attenuates greatly through ontogeny. So we want to pick, it's key that we just use the second and third year of life because at that point they're still growing fast. Second year is probably actually the best. And then we measure the oxygen isotopes here and confirm that these two bands are in fact winter. Remember those are positive values. That's telling us this is the winter growth cessation. And then we count the bands between here. We tried using dendroclimatological software. Instead we ended up just image J in our eyes now. And it's time consuming, but we got a good record going from almost really, well, we went quite to Seattle in the literature all the way up to Kodiak Island in her initial master's. And what we found is there is a nice correlation that as you go further south and you have a longer growing season, there's more tidal growth bands evident in these shells. That's not too surprising. What we were mostly interested in, though, is how well we can tell this, like how much variation was there within a population. And that was where the news is kind of bad. And what we found, like here, just zooming in just on Kodiak Island, these are 4 different locations in different parts of Kodiak Island. We have considerable variability in the number of increments grown in these different populations. So this proxy's problem is that we're probably only going to be able to detect really massive changes in seasonality. If we're going to change over a month or two, we're probably going to miss it. If we're going to change 3 months, we'll probably see it. So we're going to have to couple this to oxygen isotope records. So that's why we were just up in Dutch Harbor. So here's Christine. She was doing a lot of diving up there, and so she dove last year and deployed a bunch of data loggers, salinity and temperature data loggers in various locations around the Dutch Harbor area. And then we came back this year to harvest some clams and collect the data from the loggers. We just got them back. In fact, we're still just doing the raw processing on the data. But the idea is we're going to build our own calibration equation for oxygen isotopes here. We spent a ton of time sampling all the freshwater sources on this island. There's no big rivers, but there are a few small rivers, trying to get an understanding of the variability of the oxygen isotope composition of the water. So if we can try and couple a length of growing season record to at least a qualitative measure of temperature, coupled again to the paleobiogeographic distribution of these seal species we see in archaeological and fossil deposits, we hope we can gain some insight into whether or not these other studies are onto something and sea ice actually came this far south. We're skeptical of it, but it is possible. We did confirm that these were in fact ring seals that were present in these sites. So hopefully this will come out soon. And before I get into this interesting side question, and this is actually the anthropologist in me, I can't get beyond this idea that nowadays people die if they eat these clams, and yet that was one of the staple diet components in the past. And I can't help but wonder if that's got to do with anthropogenic causes. Increased dinoflagellate populations in certain of these areas. The locals, the Sea Grant there blames it on warming, that it's been getting warmer and warmer and warmer here, and you have a greater concentration of saxitoxin in many species. It's starting to affect the puffins, apparently, even. It's so bad. I would love to find a biomarker for saxitoxin that I could measure from some of these bay floors. If anyone knows that off the top of their head, a way to make that measurement and get a handle on how people avoided dying. There's an alternative. It could have been that there's just as much saxitoxin in the past and they only ate them in winter, which would give them time to flush more of the saxitoxin from their system. Or, sadly, they fed them to their dogs and watched their dogs for a day before they ate them as a way to test. But there's not too many dogs in the Aleutian, so any insight that you guys can give me into what causes saxitoxin concentration, there's not a whole lot of literature on it in terms of its sediment biomarkers, unfortunately. All right, last episode. How am I doing on time? 45 minutes in? Okay. All right, this is going to be the last story, and I've got a show and tell item here. So this is an oyster it's like the umbo or the chondrophore section of a Cresostria virginica oyster. And this project I'm about to describe is still underway. We're about to write the first paper. And the idea came from Rowan Lockwood at William and Mary. And what she's been on a series of papers lately, looking at this freaky size distribution of oysters through time. And so that one I'm showing you is nowhere near the biggest oyster we've gotten. So that's about a 125,000-year-old oyster. It comes from a fossil deposit on what's now the shore of Chesapeake Bay, but it sure as heck would not have been the shore of Chesapeake Bay 125,000 years ago. And the oysters you sometimes see there are gargantuan. We've got some so big that I can't even embed them in epoxy. They're the size of a skateboard. And not only that, they grow strangely. So if you look at an oyster that grows around here, they thicken primarily at the hinge. These guys thicken the whole shell. So the left valve of these guys will be 2 and 3 inches thick. The shell will be this big. A single oyster would have been a comfortable meal for a person. Absolutely just freaky. They're not all like that, but a lot of them are like that. And that's intrigued Rowan. I'm going to have some data plots in a minute talking about her particular shells. This is one of the fossil deposits, again, trying to dispel this notion that fossils are rare and difficult to find. I mean, I don't care who you are, just cruising by here in a John boat, you're going to say, wow, what's the deal with that 2 or 3 meter deposit of oysters? And if you dig, they go deeper and deeper and deeper. And this is a great place to preserve oysters. This is an example of a fairly large oyster that came from here, this particular site. This is a modern oyster living not far from it today. So this would be like, you know, the oyster you eat at a restaurant right now. This is only the hinge area of its earlier predecessor. If you break it down, so the main question is, can we use oxygen isotopes to figure out how old these guys are? Because what Rowan's interested in is, did these ancient oysters grow really fast? Or did they live really long? Or did they do both? And then on top of that, if they do prove to be significantly different from the modern oysters, why? Why were these oysters so giant then and so comparatively tiny today? Same species, it seems. The big problem is we don't really have a great way of measuring how old an oyster is by looking at it. And I'll talk about some of the few attempts in a minute. But if we assume that size has something to do with age, and that's what Rowan and the guy Tobin Rick and a few others did in this paper, and you compare modern oysters in Chesapeake Bay to colonial period, this is like from Jamestown, a few of those archaeological sites, to the Pleistocene, this is Ice Age and earlier oysters, what we see is that probably, you know, I keep doing this, you have a great deal of overlap in size. But none of the modern oysters approach the size of the ancient ones, even the archaeological ones. And the reason they're focusing on the archaeological ones, and some of these are even pre-European contact, is this idea that maybe it's overfishing and the way we've just trashed Chesapeake Bay over the past few hundred years. That back when the bay was pristine, back before industrialization, oysters grew bigger. They didn't. It didn't seem. So this seems to be something unique to the Pleistocene. You get these mega oysters like this. So she wanted to figure out, okay, the size, mean age, and how. And so there's like 3 papers that address this, and I happen to have written one of them, and that's how she found me. I didn't know Rowan. She just emailed me out of the blue. One was a paper, probably the first and best paper in this, is Kirby et al., 98, looking actually at some Gulf oysters. And they were saying that these oscillations you get along the chondrophore, that's meaningful for time, that you can age an oyster by that. Well, this is one of the Pleistocene oysters. We can't even detect that. It's just not, we don't get oscillations there. Donna Surge had done a study from some Florida Gulf oysters, modern oysters, and she had noted these chalky layers and the dark gray layers, and in her case, they didn't seem to correspond to anything. It was just something skeletal perhaps, but didn't correspond to any environmental or seasonal variation. I studied these in the Georgia Bite, and it was a crappy paper because I was using a bad isotope system, and I was only had like 1 measurement per layer. But what I showed is that many of the gray bands were winter in Georgia, but not all of the gray bands were winter in Georgia. And this is what we see in Mercenaria, where you have, depending on where you are latitudinally, they're going to grow different bands in different times of the year. And it might even vary in one place over just a decade or so. So in other words, these bands is not like tree rings. They vary through space and time. So she thought isotopes are going to be her best method for figuring out the ontogenetic age of these shells. So she came to me because I'd done a ton of this work, not for paleo work, but to understand when humans used to consume oysters in different parts of the world. I do tons of what's called season of capture analyses, where I will measure oxygen isotopes through the ontogeny of an oyster and look at the sinusoid and look at the terminal marker of the sinusoid and figure out, was this oyster collected in winter, spring, summer, or fall? And that's useful to archaeologists for a number of reasons I won't go into today. But using this for a different reason, we tried to measure as much as we could through the entire expression of growth. And we again revealed these sinusoids, although you're going to notice they're very sawtoothy. And what that tells us is they're not continuously growing. So when an organism grows and water temperature is fairly conservative, you generally have a smooth sinusoid of oxygen isotopes. If an organism stops growing and you truncate the most positive isotope values in that smooth sinusoid, what you get is a sawtooth sinusoid, what we call a cyclosoid. So whenever you see a cyclosoidal pattern in an isotope profile, you tend to think you're looking at growth cessations. And based again, the more positive values indicate colder waters if we assume constant delta H new water. What this is meaning is that these guys, 125,000 years ago, were not growing throughout the winter. And many times when they would stop growing in the winter, they would precipitate this sort of darker, denser shell material. And that's what's depicted in these vertical bands right here. But again, it's kind of like what I think I saw in Georgia during my master's research for archaeologists, in that you also have a lot of bands that don't seem to have anything to do with season. And some oysters, they seem to behave themselves really well, and the banding does have a lot to do with their ontogenetic age, and other oysters right nearby don't. So oysters are a little bit of an ugly organism, but nevertheless it's working for what Rowan wants to do. So she can now look at one of these shells and age date it. Not geologically age date it, but ontogenetically age date it. And what she's finding, at least with the small sample sizes we've gotten so far, is that these oysters don't seem to grow that differently. They just live a hell of a lot longer than modern oysters do. There's another team working on the same issue up at Cornell, and they're focusing on more southerly fossil deposits, South Carolina, and we've measured a couple from South Carolina, North Carolina. What's interesting is they don't ever find the big old oysters in South Carolina 100,000 years ago or more. They all look just like modern day oysters, same size, same everything. What's intriguing is why? And this other reason Rowan came to me is her first hypothesis of why we had these monstrous Pleistocene oysters and no monstrous modern oysters is because of the ultimate oyster predator, which is us. And that as soon as humans arrived in North America, they started eating the hell out of oysters, and they always have. But this doesn't add up. Humans weren't in the southeastern U.S. 125,000 years ago, yet their oysters seem to have been limited in their size and age, kind of similar to today's oysters. Now she's sort of focusing more on this explanation of a different predator. Perhaps limiting the lifespan of these oysters. She's in the earliest stage of this research. The paper we've got forthcoming is really just making the argument that we can use oxygen isotope cycles to understand ontogenetic age in oysters. Unfortunately, because we're not going to be able to confidently use banding or the Kirby method of these oscillations in the we're never going to get very large sample sizes, because to generate these data, that's 3 people working for 7 days. Per shell. And if you were to pay a lab to do this, this would be $2,000 or $3,000 worth of analyses to get this many samples done. So you can't expect to get large N's here. So we're working up there. We tend to think we're getting some intriguing quasi answers, but the method was useful. What's my big take-home message here? I probably bored you to death by now. Here's what hopefully you'll remember 10 years from now, is that this obscure science of the skeletal growth and chemistry of marine organisms is actually an essential component of paleoclimate research. And it's been used to explain everything from the way our geometry to the sun changes over hundreds of thousands of year time intervals to understanding perhaps the particular tide at which a Paleo-Indian collected a clam in Alaska. It's a really remarkable tool, even though it seems rather obscure. My final caution for the students in here is this. We always talk about how great interdisciplinary research is, and it is. I mean, I'm like the poster child for that. But interdisciplinary research is really dangerous and rich in pitfalls. And I'm sitting here talking to a room full of marine biologists and ecologists, and I would bet, and I would hope, that half of you have found big mistakes in things I've said. And just like if you were to come and talk in my department about the geochemistry of skeletons. But the thing is, we've got to do this. And we got to build teams to do this. And that's why I've been fortunate to work with Ruth and a lot of other marine biologists around the world, archaeologists, geologists, paleontologists, climatologists, even climate modelers. That's the real key. Our life is too short to become an expert in maybe more than 2 or 3 things. So you've got to build useful teams where you can communicate to each other. Anyway, I'm off my soapbox. Thanks for having me.
All right, thank you, and we're going to ask that you use the microphone for questions. So does anybody have questions?
Wow, it was the perfect talk. I explained everything.
It's a little question.
Sure.
Early on, I can speak loud enough.
Okay.
Oh, okay. Early on, you had in one of your introductory slides a Delta 47. What
Yes.
does that stand
That's
for?
the abbreviation for clumped isotopes. And 47 is the abbreviation for what are called clumped isotopes or isotopologues. If I can make a plug here, so in my role as department chair, one of the big things I do is try and facilitate grants for my faculty members. And I got a faculty member now who's trying to write an MRI to measure that metric you're talking about, if I ever get back to it. And so you need to buy a very special mass spectrometer that only Thermo makes. I think there's one more competitor now. It costs about $1 million. There's about 12 labs in the world that measure clumped isotopes presently. We want to be another of those. And what we have to do on campus when we try and write MRIs is get interdisciplinary work. So like we're going to be doing an electron probe MRI later, and we're going to work with a bunch of engineers who use these for compositional analysis and whatnot. We can't find a partner in another department that cares about clumped isotopes, even though the machines are that expensive. And we've been racking our minds to come up with any reason at all a biologist might want to know the temperature at which a skeleton formed. Geologists were fascinated by it. So it's kind of a frustration point. If you want to read about clumped isotopes, John Eiler's got a few papers. They're really dense and complicated and very mathy. But there's been a few, particularly a guy named Ben Passy, who just started at Michigan, used to be at Johns Hopkins, wrote a really nice summary of the applications, focusing on marine organisms, but focusing on extinct marine organisms. I just don't know of a bio application yet to this exotic measurement.
There's also a couple of websites now that you can find where they've attempted to simplify, I think for students, to simplify the explanation. So those are pretty easy to find if you're interested in that topic. Okay, couple quick things. There are still spots available at 4 and 4.